Yeah, do we put
Yeah.
that with the two end results on the right-hand side of the square?
No.
No, so then you add up the two parts, the total. So you add the 2 and the 4.
Okay.
Yeah. And I'm going to go over it in a second, piece by piece. So, let's see how.
Yes.
And
And you have 2 out of 6.
okay. Times 100%, right?
There you go.
Please, Patrick, Henry, can y'all see the problem?
Yes.
Yeah.
Making sure. Yeah, it's really good to like just get in the habit of practicing these before I go over it, because you're for sure going to see one of these on an assignment and probably the exam. So make sure that you're doing these practice problems in the morning, because it gets easier as you do it, and then it'll be like. Tom, do you think they need a few more minutes, or do you want me to go ahead and start going over the problem? Is everybody there?
I think you start.
Okay. All right. So for the practice problem this morning, the question was what percentages of corn and soybean meal would be needed to achieve a ration with a crude protein content of 20%. So I gave you the numbers. So let's say corn that you're using has 16% crude protein, and the soybean meal you're using has 22%. So how do you set this up? So the numbers that go on the left are, of course, your ingredients that you're going to be using. And then which number goes in the middle?
20.
20, right. So what you want to achieve. So how do you use the square? How do you get the numbers on the right?
You subtract.
Right, subtract. So you would get 2 parts corn. Is that what everybody got?
Yeah.
Okay. And then how much soybean meal did you get?
4.
Right. Perfect. So then you add up those 2 parts. So your total would be what?
6 parts.
Right. Correct. Okay. So then if you just need parts, you can stop there. But the question, and most of the time, you're going to want to take it out to a percentage. So how do you get the percentage? If you have 2 parts corn, how do you convert that to a percentage?
2 divided by 6.
Yep. And then you can multiply by
100.
Yeah, correct. So that gives you roughly 33.33 or 33, depending on how you round. So just make sure when you round that you're rounding, correctly for however many decimal places you want. And so then for soybean meal, what percentage would you get?
66%.
Yes, correct. So I just took this out to 2 decimal places just because, but you could leave that at 33 and then that would be 67. So however you round, you just want your total to be 100%. So that's just a good way to check it. So does anybody have any questions on the Pearson square or this example before we start the lecture for today?
Okay, so how do you,
So it's 2 divided by 6 times 100.
I used to say 2 to 6 is 2 to 33. 7 and a third.
2
1/3.
2 divided by 6.
And 4 6.
Divided by 6, and that's going to give you in your calculator 0.33333. Multiply that by 100, and then that's 3%.
Is that all good? Anybody have any other questions?
Divide that into 100.
So everyone is good on the Pearson square? Feel like you get it, or do you want another example like on Thursday?
I think we can do another one Thursday just for fun.
Okay. I like Pearson School, so I can do more things. So, all right. Well, if everybody's good on this today, then we're going to go ahead and move on to the lecture. If anyone has any questions.
I just had flashbacks. I mean, you know, was for me, or the cookie store was yeah, it'd be Iraq for y'all now. It's just order on all y'all classes.
All right. Can everybody see the lecture slides?
Yeah.
Yep.
Okay. So today we're going to talk about animal genetics and breeding. So kind of another really important area of animal science since we've already talked about nutrition. And then on Thursday we're going to talk a little bit about physiology of reproduction and how that works. So today we're going to discuss genetics and breeding. So there can be very, very big impacts of genetics on livestock. So genetics can lead to huge changes in the efficiency of animal production. And that's mainly due to the use of quantitative genetics, which I'll explain a little bit later, that is used to improve the genetic merit of cows. It can also be improved nutrition, improved animal health, improved management practices, availability of bovine somatotropin, which is also growth hormone, and then the widespread use of AI that allows farmers to purchase semen from superior sires. We already talked about advantages of that. So those are all different ways that livestock can be improved, and genetics is a big part of that. So just for an example, in 1945, it would take approximately 3 months or 13 weeks to grow a 2.6 pound chicken with an average growth rate of 0.1 kilograms. So today, it only takes 8 weeks to grow an 8.1 pound chicken with an average growth rate of 0.45 kilograms per week. So that is a huge, huge improvement in poultry, and there's also been many improvements in other species, but that's just an example to show you how genetics can really impact livestock. So the vast majority of this improvement is due to the effect of breeding or genetics. So very important. So just to kind of get you familiar with some of the terminology before we jump in to genetics. So gene is the unit of heredity, and so that encodes a single protein. And you'll kind of see how all of this fits in a little bit, but I just want to get you familiar with the terms. Allele means a specific locus on a chromosome that usually encodes a gene. And a locus is basically just a particular location on a chromosome. So again, my weird memory things, locus, location, kind of how I keep that straight. And then chromosome is a long DNA molecule that encodes multiple genes. So those are some kind of like little terms about kind of things to do with the chromosome. So these are just some other terms that you'll encounter when you're talking about genetics and breeding. So epigenetics is basically heritable changes in gene expression that are not encoded by DNA. So it's pretty much anything that changes the way a gene is expressed that's not actually encoded by the DNA. So this could be caused by the environment, age, disease, a number of different factors. But so that's an important term to know there. Epistasis is just when two or more genes interact. That's just what that term is called, or that interaction is called. Gene expression is the synthesis of RNA from the DNA template. So if you think back to that biology lecture where we talked about how the DNA is housed in the nucleus and then the DNA is converted to RNA, and does anybody remember what that's called? Is it transcription or translation? Transcription, yep, correct. Because you're just going from DNA to RNA. You're kind of saying similar, so you're just transcribing it. You're not completely changing it yet. So very, very good. I'm impressed that you remembered that term. And so then genotype is basically just the full collection of the genes or the DNA in a specific individual. So just to go over a little bit about the fundamentals of animal genetics and genomics. So heredity, which we've kind of mentioned heritable traits a little bit before. Heredity is just the transfer of genes, and so genes are responsible for different traits or characteristics from two parents to the next generation of offspring. So it's just the transfer of that DNA, basically. And then genes are the unit of inheritance, so that's how things are inherited, is through genes. And genes are arranged along chromosomes in a specific order. And then DNA is the chemical coding for the genes. So that's kind of a lot of explanation. So I have this picture to kind of help show you how that's kind of broken down. So you have your cell, your animal cell, and you have a nucleus, because remember the nucleus is where your DNA is. So in the nucleus you have your chromosome. So that's basically kind of X-looking thing would be 2 chromosomes right there that are joined in the center. And so then on the chromosomes you have genes, which are lined up all along the chromosome, as you can see in the picture. And then in the genes, you can see the DNA. So basically, the DNA makes up the genes, which make up the chromosomes, and then the chromosomes are housed in the nucleus. So that's kind of how that structure works. And then we're going to talk a little bit more specifics about DNA and everything in a little bit, but that's how that looks. So when I'm talking about chromosomes or DNA, you kind of understand where they're coming from. So does everybody kind of understand what makes up what and how this picture kind of works? Do you need a little more explanation or anything? Okay. Just let me know if you have any questions, because it's heavy information. So the cells can have what's called ploidity, which is basically just a term for kind of how many chromosomes do they have. So the main types are diploid or haploid. So the majority of cells are diploid, and so if you look, it starts with di. So if a word kind of starts with di, what does that kind of tell you it might mean? Two, right. So diploid has a pair of chromosomes, or pairs. So they have sets of 2. And then an exception would be gametes, which are like your sex cells. So those would be haploid, and they only have a single set. And that's because when those join, because in order to have a fetus, you need an egg and a sperm. Each of those have 1, because when they join, they're going to form 2. So you want to end up with 2, so you don't want them to have 2 already, because then that would cause problems. So that's why they're the exception to the rule, and they have 1, because they're going to join. So when a male and female gamete unite, a zygote is formed, and that zygote will carry genes from each parent. So they'll have, 1 set of chromosomes in each one, and then that'll come together, and then that zygote or fetus will have a pair of chromosomes, so 1 from the mother and 1 from the father. So that's how that works. So the number of chromosomes that we have varies with species. So different species have a lot of different ones. I'm not going to go into how many each species has or expect you to know that, but just know that it does vary with species. So the genetic code consists of a series of codons, and a codon is basically just a sequence of 3 nucleotides, which I'll explain in a little bit, that corresponds with a specific amino acid. So DNA is really, really important. That's like what all of our genes are made up of. All of the chromosomes essentially are made up of DNA. And that's what codes for pretty much everything about us. So DNA is also known as deoxyribonucleic acid. So you can kind of be familiar with that, but everyone is familiar with the term DNA because deoxyribonucleic acid is it's a mouthful. So its structure is a double helix that's composed of 2 strands of alternating deoxyribose, which is just a sugar, and phosphate. And I'm going to show you a picture in a second to kind of show you how this looks. And then DNA is made up of what are called nucleotides. So they're the basic building blocks of DNA. So like I said, it consists of a sugar, which in DNA would be deoxyribose, and 1 of 4 bases. So the bases in DNA are adenine, guanine, thymine, and cytosine. And those are divided in two categories basically based on their structure, kind of, whether they're purine or pyrimidine bases. So your purine bases would be adenine and guanine, and then your pyrimidine bases would be thymine and cytosine. And for this class, I'm not going to really get into what distinguishes those from each other because that's kind of a little bit more involved than you need to get at this point. But just so that you know that they are classified that way. So to kind of help you understand what I just said on the last slide, I have this slide with the structure. So if you look at the key over here on the left, you have different bases, 4 bases of DNA. You have your adenine, thymine, guanine, and cytosine. And so make sure that you are familiar with those. Those probably show up on an exam. It's important to know the 4 bases that make up DNA. So go ahead and familiarize yourself with them. So how this works is that you have a nucleotide, which consists of your base, that sugar, and then a phosphate group. And so you have just a bunch of those. So you have this sugar backbone, and then you have your bases that are joined in the middle. So in DNA, cytosine is always going to be paired with guanine, and adenine is always going to be paired with thymine. So you're always going to have a purine base paired with a pyrimidine base. Does everybody see that? So adenine, if you can see like the different, adenine's going to be paired with thymine, and then cytosine with guanine. So that's always going to happen. So how these are arranged, you know, depends on your genetic code, but those pairs will always be the same in everybody's DNA. So C to G and A to T. So then once you have that structure, you have this DNA ladder. So this blue would just be those sugars and phosphate groups. And then that structure is twisted into what is known as the double helix. So just think about if you kind of had something like a ribbon or something straight and then you just twist it, you're going to kind of get that double helix looking structure, that curve. So that's basically how DNA is structured. And the bases are held together by hydrogen bonds in the middle. Does anybody have any questions on the structure of DNA on this slide? So the inheritance of traits is determined by a number of different factors. So it's determined by the sequence of the DNA, because remember I said you always have those same pairings, but the order that they might be in is going to vary. So someone may have like an A and a T and a C and a G, and someone else at that same spot may have 2 ATs. Does that make sense? So the sequence that those pairs are in is what makes up your DNA. Another thing that can influence how genes are inherited is modifications of DNA, which I'll talk about a little bit more specifically in a little bit. But basically the DNA can be modified by the addition of other molecules or other carbon groups, things like that. So methylation is just one example. So another thing is that there can be modifications of associated proteins or histones that can change the activity of a gene. So a histone is basically just a protein that packages and orders DNA. Because you've got these long strands of DNA, and it's got to be packaged into a chromosome in a cell, which is, you know, pretty small, and you've got these long strands that need to be organized. So if you look at this picture, the histones would be these kind of pink circular molecules. And so basically, if you think about it kind of like a fishing reel, you've got your DNA line, and then you wrap it around that histone, just like you would wrap your line around a reel. And so that keeps it organized, and in that particular histone, you've got a lot of DNA, and it allows for more DNA to be compacted into that chromosome. So that's pretty much all that histones do. But histones themselves can be modified, which can affect how the DNA is then transcribed. So does everybody understand histones? Does that make sense? So there's been tremendous progress sequencing the genome of domestic animals such as cattle, chickens, horses, and pigs. And comparative genomics is basically just comparative, so it's comparing the genomes of different species. So that's a term that you'll probably see if you say in animal science, so be familiar with that term. It's bold and underlined, so that's kind of a giveaway. But it's pretty easy. Comparative genomics is comparing the genomes of different species. So pretty easy to remember. And so just kind of things that have to do with comparative genomics. Closely related species would obviously have more similar gene sequences along a chromosome. And then the more differences that you see kind of tells you that the more the species are more distantly related. So that makes sense if you think about it. So like if you have any siblings, your gene sequences would probably be more similar than they would be to like your cousin. So that's kind of how you can think about comparative genomics in different species. So there can be genetic mutations. And so genetic mutations basically just means any number of changes to a genome of an animal that can happen. So changes can sometimes be really harmful, and sometimes changes actually have no effect. So it just kind of depends on which gene is involved and what is happening to that gene. So there are many different ways that genetic mutations can happen. So some of these mutations could be the deletion of a gene entirely, or part of a gene, which is going to cause that to not function properly. And if it's not there, then it's really going to not work. Gene duplication. So you could have a gene that is somehow duplicated twice, and that could cause effects. Gene inversion. So inversion basically just means that if you have kind of one gene on top of another, they might be inverted, and the order might be switched. Gene translocation. So a gene could be moved to another location on a chromosome, and it may not be in the same order anymore, so that would really affect it. And then you could have a point mutation of just a single nucleotide. So you could have one nucleotide in that DNA structure that is mutated, that's not correct, and that will really impact how that gene is going to be transcribed and how that trait is going to be expressed. So like we talked about earlier, I mentioned epigenetics. So epigenetics is just heritable changes in DNA expression that are not encoded by the DNA. So epigenetics is anything that happens to that DNA that is not actually encoded in the DNA itself. So that would include covalent modifications of the histones, like we talked about, the protein structures that the DNA is wrapped around. And it can include methylation. So methylation is just adding a methyl group, which is just CH3. So if that methyl group is on there, that can change how that gene is going to be transcribed. And then epigenetic effects can also be induced by environmental factors. So for example, if the dam or the mother is very malnourished during gestation, that could lead to defects in her offspring, because that could impact how the genes are being passed and what's happening to her genes, and then how that's going to impact future offspring. So it's really, really important to realize that many things can affect how your genes are transcribed. So lifestyle factors. I know I heard one time that mothers that were pregnant during Pearl Harbor that were there were so incredibly stressed that the babies that they then had were more prone to stress in their lives just because their moms experienced Pearl Harbor while they were pregnant. So that would kind of be an example of epigenetics, and it's something that happened outside of the DNA that influenced the DNA. Does that kind of make sense? Good. So another part of the chromosomes is the telomere. So the telomere is just multiple repeats at the end of a chromosome. So at the end of the chromosome, you want to stabilize it. So the telomeres just stabilize the chromosomal ends, and so they also protect the genes in that region near the end of the chromosome. So they're very important because they kind of keep everything in place and stabilize the chromosome itself. And then like you have genes down at the bottom close to the end, and you want to make sure that those are protected. So those telomeres are kind of just like almost caps on the end of the chromosomes, if you think about it that way. And so telomeres are kind of interesting because telomeres will actually shorten with progressive cell cycles. So we all know like cells divide, especially during growth and development, things like that, cells divide. And so as a cell divides, the telomeres start getting shorter. And it's actually been figured out that shortened telomeres can be related to aging. Because as you age, your cells will have gone through a lot of different cycles, And so to kind of show you how that looks, on this picture, you have a telomere right here, this dark pink, and then you have your chromosome. And so then as you go through these different cell divisions, you can see what happens to the telomere. It's getting shorter and shorter and shorter until by the end it's almost non-existent. And so when you don't have longer telomeres, those genes that are at the ends of the chromosome are going to be a little bit more prone to you know, things happening to them because they're not exactly protected anymore by that long telomere. And so basically all of that can kind of be associated with aging. So there are different types of gene action, so how genes can actually act. And so chromosomes and genes are homologous pairs. So homologous means the same. And then genes can be additive or dominant, or partial dominant, or overdominant, or recessive. So I'm going to explain a little bit about what that means. So if genes, you may have like a trait that is influenced by two different genes, right? Like there's two potential genes that you could have. So they could be additive. So you might have, if you get both of those genes, you have like an additive effect that's better than just two of the same or something like that, or one. Or they could be dominant. So we'll talk about that a little bit more in an example. But for example, like brown eye color is more dominant than blue eye color. So if you get the gene for brown and you also get a gene for blue, you're going to have brown eyes. That's kind of how that works. And I'll explain that a little bit in a couple slides. They could be partially dominant or over-dominant or recessive. So recessive would kind of be that blue eye trait. So recessive would mean that it's not dominant, so you would need to have all of those genes being for blue eyes, basically. So homozygous is basically just a genotype that consists of two identical alleles at a given locus. So remember, locus is just a location on the chromosome, and then alleles are a particular locus. So if a genotype has two identical alleles at that point, that's homozygous. So two of the same. And then if a genotype has two different alleles at a locus, it's heterozygous. So make sure you keep that straight. Homo is the same, and then hetero would be two different ones. So those are just two different genotypes that you could have. So this will probably make a little bit more sense once we get to kind of the example. But there are 6 genetic combinations possible for a single person. So homozygous and heterozygous, for each trait, you would be either homozygous or heterozygous for a particular trait. So you could have, like if you have, a dam and a sire, one of them could be homozygous for that trait, and the other one could be homozygous for that trait. You could have both of them being heterozygous. You could have like the mom is homozygous and the dad is heterozygous. So I'm not going to kind of read all of these because it's just going to kind of sound a little bit overwhelming. But basically for each trait, the mom or the dad would be either homozygous or heterozygous. They would either have the same genes or two different ones. The same alleles or two different ones. And so there are lots of different ways that these genes can be combined. So this might make more sense in this example. So it's a square because I've figured out that people in animal science really like squares to do things. You have the Pearson square, and then in genetics you have what's called the Punnett square. And so basically this is just a really simplified version of how to predict the outcome of a particular cross or breeding experiment. So, for example, you would have the genetics of parent 1 up here, and you can switch. It doesn't have to be 1 on the top and 2 on the side. It doesn't matter. And then you have your genetics of the other parent on the other side. So this capital letter and lowercase letter are different. So would this parent be homozygous or heterozygous? Heterozygous, right, exactly, because they're different. So this parent would also be heterozygous. So this would be a heterozygous times a heterozygous combination, like we talked about on this slide. So how the square works is basically you just bring down, this first square is just you bring down that first letter and then you bring that other letter across. So that's going to give you BB, both capital. And then for this bottom square, you're going to bring that big B down and then little b across. And then same thing for this other one. So this B is going to go across, little b is going to come down. And then same thing for the fourth square. You're going to bring this little b all the way across and then little b all the way down. And we're going to do an example in a second to kind of show you how that would work and why that would be important. So this would be an example of polled cattle. So does anybody remember what polled means from the first section?
It's like dehorned, isn't it?
Dehorn, right. So they don't have horns. So we're going to use this example to kind of show you how you can use the Punnett square based on the parents to determine what the outcome of the offspring, what the chances are that they're going to have horns or that they're not going to have horns. So for this example, let's say that polled is the dominant trait. So polled is going to be with a capital P because that's dominant. And then the horned trait is recessive. So we're going to denote that with little p. So for this example, that's what we're going to do. So a polled cow would be either homozygous for the dominant trait, so they would have PP, like big P, or they could be heterozygous for the dominant gene, and that's because polled big P is dominant. So if it's dominant, you only need one. So if you have big P, little p, it doesn't matter that that little p is there. That makes them heterozygous, but that big P is still what's going to control the gene. Does that make sense? So then in order for a cow to have horns, because horned is recessive, you can't have a big P in there. So you have to have little p, little p. You have to have two of the recessive in order for the cow to have horns. So that's how that works. So in this example, this first example on the left, so you have a horned mother, so she's little p, and then you have a homozygous polled father. So that would be big P, big P. So that Punnett square, this is how that would work. So each offspring would be getting a big P from the dad, but a little p from the mom. So would these be heterozygous or homozygous? The offspring. Right, because they have one big P and one little p. They have different ones. So would this offspring be polled, or would they have horns based on what polled being dominant?
Polled.
Right, so all of these would be polled in that particular cross because that's dominant. And the father only has big P's to offer. So in this next example, we have a heterozygous polled sire mated to a horned dam. So when you do that Punnett square and you carry those across, you get a little bit of a different outcome. So you have 50% that are going to be heterozygous, and then what are these at the bottom? Are they hetero or homo? Homozygous, right, because there's two little p, they're the same. So in this example, so these two, the chances of your offspring being polled would be what?
50-50.
Yeah, right, 50-50, because you have two that you have a chance where you could have 50% would have a big P, so they would be polled, but you also have 50% that are just going to get little p's. They're going to get a little p from mom and a little p from dad. You have 50% chance that your offspring is going to be horned because that's recessive. So does everybody kind of understand how that works and how it's just kind of a simple little way to determine, what your outcome of a breeding pair might be if you know their traits? Does the Punnett square make sense?
Leanne, are you going to use the word carrier?
I didn't really go into detail about carriers.
You did, but in short, if you can get your arrow to the right place, for this second of the two sets of cows here, The cow on the left is a carrier because even though it is polled, it is carrying the non-polled gene.
Yep.
So you sometimes hear when you're seeing stuff on TV, they talk about somebody that's carrying a disease or carrying a gene. That's what's happening, and that's why even though this is a polled cow, it can still have kids that have horns because it's carrying that gene, though it's not getting expressed.
Right.
Anyway,
Yep.
just
Yeah.
the word carrier, I remember that one from college. So
yeah, that's a really good point, and that's kind of also really important when you think about like diseases, genetic diseases, where two healthy parents that don't have a disease can have a kid that all of a sudden has this genetic disease, and that could be because both of them are carriers of that recessive gene, and then they can have offspring that gets a copy from each of them of that recessive gene, and then all of a sudden their offspring is going to express that, so that they're going to get that disease. So that's really important. So thanks, Tom, for bringing up that term. So everybody feels pretty good about the Punnett square?
Yes.
I don't understand how it works. Okay. So how can we apply animal genetics in breeding? So there can be different types of inheritance. So there can be what's called polygenic or quantitative inheritance, and that just means that the heritable traits are based on multiple genes. So remember poly, like polyestrous, many estrous cycles. The poly means a lot, multiple. So animal breeders employ quantitative genetics together with what's called marker-assisted selection, or MAS, to improve breeding or to produce hybrids or crosses between different breeds or different grandparent stock. So they can, if they have the genome of an animal, they can look at the different genes that those animals have and then kind of decide which ones you want to breed to each other based on what you know to kind of get a specific outcome. So like if you know if they're polled or horned and you know what their actual, whether they're heterozygous or homozygous, you can actually determine which ones you want to breed to each other based on whether you want a large chance of polled offspring or you want horned offspring. So you can use this for many, many different things. That's just an example. So when it comes to breeding, heritability is the proportion of the total variation in the phenotype that is due to the gene effects. So remember we talked about epigenetics, how there can be variation in the way DNA is expressed based on environment and things like that. So heritability is the portion of the variation that is due to gene effects, so just effects coming from the gene. So heritability can estimate, yeah, we don't need both words there, but it can estimate the degree to which a trait is passed on from the parent or offspring. So basically, how likely is it that if a parent has that gene, it's going to be passed off to its offspring? So how heritable is that trait? So for example, highly heritable traits would be carcass quality. So like in Angus or breeds that have really, really good carcass quality, those traits would be highly heritable, meaning that there's a good chance that if the mom has good carcass quality, that her offspring will have those traits as well. You can also have moderately heritable traits that are kind of in the middle. So growth rate would be an example of that. So the parent would kind of a little bit control their genes that would pass down, would kind of control a little bit of the growth rate of the offspring, not as much as it would a highly heritable trait. And then you can also have lowly heritable traits, so like reproductive traits. So things like litter size in a pig or mothering ability, things like that, any of those reproductive traits, they would be lowly heritable. So just because, one sow had really high litter size doesn't necessarily mean that her offspring is going to do just as well. Of course, you have your breed characteristics, and breeds are known for certain things, so I mean, they would still do well, but if it's a lowly heritable trait, they may not do as well as their parent did. But that could be a good thing. If it's a bad trait that you don't really want, you might want it to be lowly heritable, have low heritability. So it's just kind of important to think about when you're breeding is, what is the trait that you're breeding for, and how likely is it that this trait is going to be passed on to the offspring? So there are basically 3 different breeding systems that are used. So you have what's called straight breeding or purebred breeding, where you're just going to be basically, breeding all animals from the same breed. They're going to be purebred. It's kind of just almost like a line breeding. It's pretty simple. You have crossbreeding systems where you can have 2 or more breeds. And so the advantages of crossbreeding, which this should sound familiar to you, would be hybrid vigor. And another term for that is just heterosis. That's kind of the more scientific genetics term. But hybrid vigor and heterosis are the same thing, which we've talked about hybrid vigor before. So that's basically where the offspring is superior to the average of the parents. So you have 2 different breeds or more, and that is going to display hybrid vigor. And a good example of an animal that has hybrid vigor would be, you might remember from section 1?
Mule?
Cross between a horse and a donkey.
Is your mule?
Right. So mules, remember, they're hardier. They have better kind of disease resistance and things like that because they have hybrid vigor. So that's a good example. Another good example will be like if you breed an Angus to a Hereford and you get a Black Baldy. That Black Baldy is going to maybe have the demeanor of the Hereford cattle, but it might have the carcass quality of the Angus. So that would kind of be a good example of hybrid vigor and why people crossbreed. So another breeding system that you might use would be synthetic inbred lines for grandparent stock. So that may be based on 1 or several breeds, but those have been really inbred to kind of keep traits around. But of course, with any type of inbreeding, you have to be really careful because you don't want certain traits to kind of pop up, and you just have to be really careful there. So marker-assisted selection, or MAS, is also another tool that people can use for breeding. So that's basically the use of quantitative genetics coupled with genomics. So it kind of just gives you more accuracy in the estimation of your breeding value. So it's really, really useful for traits that are very expensive or difficult to measure. So traits that influence meat quality and animal health would be kind of very expensive because that would determine, like meat quality, it might determine how much you get for that animal. And so they can be very, very expensive and a little bit hard to measure. So if you measure them, you want to get a little bit more accurate than you might need to for some of the other traits. So single nucleotide polymorphism, or as they can kind of be called for short, SNPs, is basically, it can be also used for breeding and kind of identifying different things there. So that's basically just when a single base pair, so base pair being those pairs in the DNA structure, so like A, T, or CG, those pairs. So when you have a single base pair that's different between animals of the same species. So if they're the same species, then most of their DNA and things like that should kind of be similar. But you can have what are called SNPs, where there's a single base pair that's different. So that is actually what accounts for most of the genetic differences between individuals. Because just because you're in the same species, obviously, I mean, look at humans, you're going to look different, you're going to have many, many different traits, and so that can be due to these single nucleotide polymorphisms, where you have differences in your base pairs. So that will be increasingly used by animal breeders because obviously they're going to be selecting for certain traits. So this is a really useful tool that they can use for breeding. So just to kind of show you how genetics comes into play, so we're going to talk a little bit about the genetics of animal color. So there's molecular genetic control of coat, hair, or skin color. So color in the fur or the hair of domestic animals is due to a polymer called melanin. Remember, polymer just kind of means like long compound of similar units. So melanin is the term for that. And melanin is made in the animal from the amino acid tyrosine. And there are 2 forms of melanin. So you have what's called eumelanin, which is responsible for your black or brown colors. And then you have pheomelanin, which is responsible for your orange or red colors. So those are kind of going to determine what color the animal is going to be. So nutritional genomics makes it increasingly possible to tailor the diet of an animal or person to its genetic code. So this is actually becoming a little bit more popular, and they've been doing a little bit more work with this. Because if you think about it, you have breeds, for example, like pig breeds, you have breeds that are more likely to deposit muscle versus fat. And so those breeds, if you know that about their genomics and their genes, you can kind of feed them differently than you would, let's say, pigs that would put down more fat instead of muscle. So you can really see what their genes encode for and how they would use those nutrients and tailor the diet accordingly. So it's getting down to more specific feeding, and it would be pretty efficient if you could actually tailor the diet to meet their specific requirements based on their genetics. So nutritional genomics is kind of just another tool that's kind of coming onto the scene. Genetic counseling is also part of genetics, mostly for people. If you've ever heard of that, like some people will kind of get their DNA mapped, and then they may have genetic counseling to kind of see like what diseases they're prone to or anything like that. So in livestock, animals that are known to pass on genetic diseases are not used in breeding programs, obviously, because you don't want to pass on any kind of disease genetically. You don't want that showing up. And then also it's used in companion animals to reduce the incidence of genetic disease. So that's really, really important, because if you end up finding out, like Tom said, that an animal is a carrier of a certain disease, you don't really want to breed that animal, because you might run the risk of breeding it to another animal who is also a carrier, and then you're going to have offspring that have that disease. So it's really important that you understand how that works. So there are different animal genetics companies. So those are basically just large multinational companies that perform animal breeding. You have heritage breeds, which are the original breeds and varieties. So as opposed to synthetic breeds or varieties, you have the original breeds that you can then go back to and get the genetics from. So those are called heritage breeds, and those are commonly used for commercial poultry production. So we talked a little bit about this before with cloning, because we talked about Dolly, the first clone sheep, first clone mammal. So you might be a little familiar with cloning. So cloning or nuclear transfer is basically using nuclear transfer from an early embryo or undifferentiated cell into an activated oocyte, which is basically just an egg. And that will produce a blastocyst that will then go to term. And so those terms are kind of just reproduction terms, which we'll talk about next lecture. So the resulting progeny would be identical to the donor of the nucleus, with the exception of the mitochondrial genes in the recipient egg, because remember, mitochondrial DNA is passed on from the mom. But other than that, the offspring is going to look the same as the donor. So just to kind of show you a little bit more about how cloning works, and actually cloning, it's easy to talk about in genetics, but I think it'll probably also make a little more sense after the repro lecture. But you have your somatic donor. So somatic donor basically means that this cell is essentially kind of just a stem cell. It hasn't been differentiated into any type of specific cell, so it can be used. And then you have your egg donor. So those two will come together to form that combination, and then you'll have those cells grown in a tissue culture, and so then you will then take that embryo and implant it into a surrogate mother, and then that will go to term, and that will give you your cloned animal, which will have similar qualities to the somatic donor. So that's just kind of visually how that works. But cloning is really, really expensive, so I wouldn't say that it's used very often. But if you just have an animal that is just like record producing, you may want to invest the money to keep that animal around. But it is very, very expensive, so it's not really like a typical tool that's used, but it's something that is there. So a cloned animal will have virtually all the genetic characteristics of the donor, and cloning was developed in the 1980s to produce domestic livestock and companion animals. So it has the potential to benefit animal agriculture with improved growth, feed efficiency, disease resistance, and reproduction. But again, that's if you have the money and you want to spend the money and the time on cloning. You can also just improve your herd by typical breeding practices and management practices. So further advantages of this nuclear transfer technology would be transgenic animals, which I know is a term that you've heard before because we've been over it several times. So a transgenic sheep, goat, or cow could actually be made to produce a human protein in large quantities that could be used as a drug. So basically, you're just inserting a gene from one organism into another organism, and that's going to make your transgenic animal. So you all should be kind of familiar with that term. So that is all I have for the genetics lecture. Does anybody have any questions on it? I know that it's kind of a lot of really scientific terminology and everything like that. It can be a lot to really digest. So I recommend if you have the book, spend some time in that chapter, spend some time going over the notes, make sure that you understand it. Because repro on Thursday is also going to be pretty heavy in the scientific terminology and the biology. So make sure that you're keeping up and that you're going through your notes. Any questions on anything? All good? What?
How's Trigger doing?
Oh, he's good.
Oh, good.
He's going as usual. Right. Well, everybody have a good and safe.
Happy Halloween.
Yes.
Happy Halloween.
Oh, Keystone people, before you go, I can't remember if I told y'all yet, the Introduction 242 class is going to happen in the spring. So feel free to go ahead and sign up for it. Go ahead and sign up for it anyway, and sign up for it whether you want it or not. Sign up for it. I just have it one way or the other. Anyway, I see some of you some of you tomorrow, Thursday.
One, I had planned this following through, so that.
I just know my boss is gonna ask me.